So hello students and welcome back again to another video tutorial by Genomics.
Homologous re-
In this video tutorial, we would be talking about how the recombination is going to take place, in what manner the recombination is going to take place, and which is the molecular mechanism involved in the process of homologous recombination. What is the molecular mechanism underlying the principle of Holliday junction? Holliday junction, which was referred to as the exchange of chromosomal fragments in between the two sister chromatids, in between the two non-sister chromatids of the homologous chromosomes. Then how homologous recombination was taking place? Please have a view.
recombination is the exchange of similar DNA sequences between different DNA molecules. Cells use homologous recombination to introduce genetic variation, as well as to repair damaged DNA. When you have completed this exercise, you should understand the double-strand break repair model of homologous recombination, understand how this mechanism differs from the Holliday model, and understand the cellular functions of homologous recombination.
We must know about certain things. We must know that how does homologous recombination take place, in what manner the homologous recombination is going to be arranged, and in what process the homologous recombination occurs in a cell. So what is this homologous recombination? Homologous recombination refers to the exchange of genetic material in between the non-sister chromatids of the homologous chromosomes. It occurs with the help of certain protein families, and those are referred to as RAC-BCD pathway, and another one is Ruvc protein. Proteins involved include RAC-BCD pathway, BCD proteins complex, along with RAC-A protein, RAC-A, and then there would be Ruv A, Ruvb, and Ruvc proteins. And Ruvc proteins. So all these proteins are going to participate together with each other and will lead to the formation of homologous recombination. Clearing all the drawings, moving forward.
The mechanism of recombination in E. coli is called the REC-BCD pathway. This mechanism closely follows the double-strand break repair model of homologous recombination. Therefore, the REC-BCD pathway serves as a good model for recombination in both prokaryotes and eukaryotes, although the specific proteins involved differ from organism to organism. In the REC-BCD pathway, homologous recombination is initiated by double-stranded breaks in a single DNA molecule, in contrast with the Holliday model, where recombination begins with single-stranded nicks at identical positions in each DNA duplex.
Now, the process of homologous recombination states that it was Robin Holliday who has given the model of homologous recombination. But according to the Michelson and Redding modification, there is no endonuclease which is present in the cell that could effectively, specifically cleave the DNA molecules at alternate sites on the same strand of the DNA molecule. Okay, at alternate site on the same homologous chromosomes. Now, single stranded nicks are being produced in both DNA molecules. Once the single stranded nicks are being produced, then the process of Holliday junction takes place. There is no restriction enzyme, there is no endonuclease which is present that can has the ability to cleave the DNA at complementary sequence on the same homologous chromosome, but there can be any restriction endonuclease that could make a double strand break in the chromosome. So this is referred to as double strand break homologous recombination model.
In the double-strand break repair model of homologous recombination, the DNA is then processed to generate a gap in the DNA with a 3' single-stranded overhang or tail. In E. coli, the REC-BCD enzyme processes blunt breaks in DNA molecules to generate such tails. REC-BCD is composed of 3 protein subunits that are the products of the REC-B, REC-C, and REC-D genes.
There are three protein subunits which are present. One is referred to as RAC B, another one is C, and the third one is referred to as RAC D. So these are the three products which are present. One is referred to as the RAC-B, RAC-C, and RAC-D proteins. This forms a complex called RAC-BCD pathway. And RAC-BCD pathway is going to move over the double-stranded DNA molecule. Now, how it is going to move to the double-stranded DNA molecule? It will migrate over the double-stranded DNA molecule, will produce a single strand overhang. How and in which manner? Please have a look.
RecBCD binds to the DNA at the site of the double-stranded break. RecBCD has both helicase and nuclease activities. It tracks along the DNA, unwinding the DNA, and frequently cleaving each strand. The cleaved DNA is destroyed.
The cleaved DNA is being destroyed with the activities of the RAC-D. RAC-D has helicase as well as nuclease activity, so it is going to move and it is going to degrade the DNA molecule.
E. coli DNA contains relatively frequent 8-nucleotide crossover hotspot instigator sequences, commonly referred to as Chi sites.
There is a specific sequence that is present, and that specific sequence which is present is commonly referred to as the Chi site, also referred to as the crossover hotspot instigator sequence called as the Chi sites. Next.
When RecBCD encounters a Chi site, its nuclease activity is altered. The mechanism of this change is unclear, but appears to be associated with either the inactivation or loss of the RecD subunit. Beyond the Chi site, RecBCD no longer cleaves the DNA from 3' to 5'. However, the 5' to 3' nuclease activity of RecBCD increases. This results in a 3' single-stranded tail on the DNA, terminating in the Chi sequence.
Okay, thank you. Now see this. Once the RAC BCD pathway is being uploaded over the double stranded cleavage, then RAC BCD moves in the forward direction. As it moves in the forward direction, it will form a single strand overhang. As it will form a single strand overhang that is going to be produced here, the RAC D activity is going to be lost after a particular moment. After a particular moment has taken place, the RAC D activity is being lost. After the RAC D activity is being lost, then the RAC B and C are the only proteins that are going to be functional. A single strand DNA is present in the form of a single stranded overhanging end which is present that has a specific site called as a chi site. Only when the chi site is being encountered by the RAC BCD protein, the RAC D activity is being lost. The RAC D loses its activity. As the RAC D protein loses its activity, so this helicase is being left. It is gone. Only the RAC B and C remains. Now the RAC B and C causes the formation of an overhanging end with the chi site present over it. And now it will integrate with the double stranded DNA molecule. How? Look here.
RecBCD helps to direct a protein called RecA to the single-stranded DNA tail. The active form of RecA is a protein DNA filament consisting of hundreds of RecA subunits, with one RecA for every 3 nucleotides of DNA.
Now, this single strand overhang of the DNA will bind with a protein, and that protein is called as the RAC protein. It has been bounded with a protein called as RAC protein. Now, this RAC protein will take this single stranded DNA molecule and integrate this single stranded DNA molecule over the complementary sequences in this manner.
The filament has 2 distinct binding sites, a primary site with the single-stranded DNA molecule and a secondary site.
Okay, it has a primary binding site, it has a secondary binding site along with itself. Now these are the two different primary binding sites that are present. Next point. There are two binding sites which are present: primary binding site, secondary binding
The
site.
RecA
Now RAC A will screen along with the complementary DNA molecule. It will scan along with the complementary DNA molecule and will try to form bonds with the adjacent nucleotide. When a complementary nucleotide will be reached the RAC protein will lead to the formation of bonds in between the invading strand and the pre-existing strand. How? Look here.
A single-stranded DNA complex participates in a search for DNA homology. The secondary site binds a double-stranded DNA molecule. Binding in the secondary site is rapid, transient, and independent of DNA sequence, meaning the filament can quickly bind a double-stranded DNA molecule and scan along its sequence.
Now, the RAC-A has bounded to the secondary site to the double-stranded parental DNA molecule, and to the primary site there is the invading DNA molecule. When the parental DNA molecule will have a complementary sequence to the invading DNA molecule, hydrogen bonds will be broken in between the parental strands and will be produced in between the parental and the invading strand. How? It is scanning still. At the place of adenine, the adjacent should be thymine.
Once a region of base pair complementarity is located, REC-A promotes the formation of a stable complex between the single-stranded DNA tail in the primary site and the complementary strand of the double-stranded DNA molecule in the secondary site. As in the Holliday model, this step is called strand invasion. However, unlike the Holliday model, the REC-BCD pathway can involve two Holliday junctions.
Now, the RAC BCD pathway may involve more than one Holliday junction. It can have two different Holliday junction that could be present. How? Look here.
Once the Holliday junctions have formed, REC-A can dissociate from the DNA. The replication machinery then fills in the gaps in the DNA.
See
This
this?
synthesis of new DNA is another feature missing in the Holliday model.
Now, what is so unique about this particular DNA model? There is a new DNA strand that has been biosynthesized. There are two Holliday models that have been produced. In the RAC BCD pathway, there was a single Holliday model. There was a single junction which was being produced in the case of Holliday model. Okay. Only one branch migration was taking place. Here from two different branch migrations can occur. Here from two different branch migration can occur in two different direction. One in this direction and another one in this direction. Okay. So these are the two different branch migrations that could take place. Looking forward.
The next step of the REC-BCD pathway is branch migration. This step is similar to that of the Holliday model, except it can involve two Holliday junctions instead of just one. In the REC-BCD pathway, this step is catalyzed by two proteins, RUVA and RUVB, which form RUV-AB complexes at Holliday junctions.
Okay. Then it will lead to the formation of the Holliday junction, and this Holliday junction will be resolved with the help of RUVA and RUVB. Now, how the resolution is going to take place, that we have to see.
RUVA protein is a DNA binding protein that recognizes the gross structure of the Holliday junction, regardless of its specific DNA sequence.
Regardless of its specific DNA sequences, it will bind to the position where the resolution has to take place.
RUVA recruits two hexamers of RUVB to each Holliday junction to form RUVAB complexes.
This
RUVB is an ATPase, and
RUVA is the first protein that would be migrating and joining to the Holliday junction. It will recruit two hexameric proteins. One is called as RUVB, and both would be called as RUVB. Two hexameric proteins are there at each Holliday junction. This RUVB itself is an helicase. It is the one that is responsible for branch migration to take place. Next point.
ATP provides the energy to drive the exchange of base pairs during branch migration.
ATP will drive the energy for the base pair branch migration to take place.
The final step of the REC-BCD pathway is Holliday junction resolution. Again, this step is similar to that of the Holliday model, except it can involve two Holliday junctions. As will be shown, the possible products are the same: patch recombination products, also called non-crossover products, or splice recombination products, also called crossover products. In the REC-BCD pathway, this step is catalyzed by the Ruvc endonuclease.
Okay, there is another protein which is going to take part, and that would be referred to as RUVC endonuclease. Now, how this RUVC is going to bind? Each Holliday junction will bind to two RUVC proteins, and this RUVC protein will either catalyze the patch recombination product or the splice. This will cleave the DNA at 90 degrees to its binding site.
For each Holliday junction, there are two possible cleavage sites. Site 1 consists of the two DNA strands that have not crossed over. Site 2 consists of the two DNA strands that have crossed over.
Now, see this. These are the DNA strands that have not undergone any crossing over. These two, second and the third DNA strand, have undergone crossover. Now, which are the different types of combinations that could be produced? It can have a splice recombination product. It can also have a splice recombination product. So it would be splice splice, patch splice, splice patch, or patch patch. So what would be the combination products that could be obtained? The different types of combination products that could be obtained are splice splice, first splice. Second is also called as splice. The next one would be referred to as splice patch. The third one would be referred to as patch splice, patch splice. And the fourth one would be referred to as a patch and patch. Patch, and here it would be referred to as patch. So these are the four combination products that would be obtained: first, second, third, and fourth. Splice, splice, splice patch, patch splice, and patch patch. These are the four combination products that would be obtained. Look here.
If both junctions are cleaved in the same way, patch recombination products result. For example, if both junctions are cleaved at site 1, patch recombination products result.
See, one thing which is very important, there is such type of resolution will not be asked neither in CSI net JRF entrance examination, nor even in the that GRE entrance examination of subjective sciences. Okay, so nobody is going to ask you how the resolution is going to take place. You just have to know about the proteins. The proteins were RAC BCD proteins, among which the RAC D has lost its activity when it was moving over the DNA molecule, when a site called as chi site was being encountered. You can look here. There was this movement
When REC-BCD
going to take
encounters
place. Just a short recap: RAC D was losing its activity when it was moving to the chi site. Look here.
a chi site, its nuclease activity is altered. The mechanism of this change is unclear, but appears to be associated with either the inactivation or loss of the REC-D subunit.
Once the
Beyond the
RAC
chi site, REC-BCD no longer cleaves the DNA from 3'
once
to 5'.
once the
However, the 5'
once the RAC DNA activity was being lost, then this chi site was the subject to the binding of the RAC A protein. How? See this.
REC-BCD helps to direct a protein
Called RAC A, and RAC A is going to bind to this existing chi site and will lead to the migration of this chi site into the other parental DNA molecule of the homologous chromosome. Okay, how? See this, it was migrating, and when it will migrate, it will find a complementary base pairing in the parental strand. It will bind to the exact nucleotide in the parental strand. It has moved, and it would be moving. So this is adenine. It will have to find a complementary thymine. Once it will find a complementary thymine, it will replace the parental strand and will bind to it. This is referred to as branch migration. Okay, see this movement has taken place: guanine, adenine, cytosine. No, it cannot find correct. Now again moving forward, thymine. Okay, it has founded a correct one. It will break the pre-existing DNA. See this. This is branch migration has taken place. Now at that towards the end, it will cause the resolution to take place. There are two Holliday junctions which are being produced, one at this particular site and another at this particular site. Thus, the rack BCD model is completely different from the Holliday model, where there is a single Chi junction which is being produced. There is single Holliday junction. Here, there are two Holliday junctions which are going to be produced. Here, there are two Holliday junctions. How? The DNA polymerase is going to repair the two processes along with the ligase enzyme. See this ligase seals the gap. Completed. This is the branch migration that has taken place. I already explained all these things to you. The branch migration, what once has taken place, then it will lead to the formation of the binding of Ruv A and Ruv B protein. Along with the Ruv A and Ruv B protein, the other protein that is going to be bounded is going to be referred to as the Ruvc.
RUVA protein is a DNA binding protein that recognizes the gross structure of the Holliday junction, regardless of its specific DNA sequence.
Next.
RUVA recruits two hexamers of RUVB to each Holliday junction to form RUVAB complexes. RUVB is an ATPase, and ATP provides the energy to drive the exchange of base pairs during branch migration.
This is where branch migration is going to take place. Next.
The final step of the RecBCD pathway is Holliday junction resolution. Again, this step is similar to that of the Holliday model, except it can involve two Holliday junctions. As will be shown, the possible products are the same: patch recombination products, also called non-crossover products, or splice recombination products, also called crossover products. In the RecBCD pathway, this step is catalyzed by the Ruvc endonuclease.
Okay. So it would be carrying out the products which would be produced by the action of the endonucleases. Next.
For each Holliday junction, there are two possible cleavage sites. Site 1 consists of the two DNA strands that have not crossed over. Site 2 consists of the two DNA strands that have crossed over.
Now, there would be cleavage taking place at site 1 and site 2.
If both junctions are cleaved in the same way, patch recombination products result. For example, if both junctions are cleaved at site 1, patch recombination products result.
This is how patch recombination products are being produced. Next.
Patch recombination products also result if both junctions are cleaved at site 2.
These are the combination products that have been produced.
If the junctions are cleaved at different sites, splice recombination products result. For example, if the first junction is cleaved at site 1 and the second junction is cleaved at site 2, splice recombination products result.
So these are the different products that would be obtained. Nothing is to be worried because nothing has to be asked about it.
Splice recombination products also result if the first junction is cleaved at site 2 and the second junction is cleaved at site 1.
So these are the different products that have been obtained. I knew that the products that have been obtained have been much more complicated in this particular process, but you didn't have to worry. You didn't have to make the products. Nothing would be asked except about the different types of protein, about the function of RAC BCD pathway, about the function of RAC A, about this function of RUV A, RUV B, and RUV C. RUV A was recognizing the Holliday junction. Ruvb was an helicase. It was responsible for branch migration, and Ruvc was the one which was resolving the Holliday junction. That's all.
Both prokaryotes
That's all about the homologous
and eukaryotes use the double-strand break repair pathway to exchange genetic information between DNA molecules.
Now,
Although the enzymes involved
try to solve the questions based upon the homologous recombination.
All differ from organism to organism. This pathway can be used in several different cellular processes. The 8 nucleotide chi site is highly overrepresented in the E. coli genome. While a given 8 nucleotide sequence should appear about 80 times in the E. coli genome, the chromosomal sequence reveals 1009 chi sites. However, DNA from another species will lack such frequent chi sites.
So there are two different types of species of the E. coli taken in this question. One which has actually 8 number of chi sites predicted. Another species has large number of chi sites, such as 1000.
Question 1. What would likely happen to DNA from another species if it entered an E. coli cell? Assume that the foreign DNA does not contain any chi sites.
See, the foreign DNA do not contain any chi site. As the foreign DNA do not contain any chi site, so what will happen? The DNA would cross over with the E. coli chromosome. The DNA would be completely destroyed by the RAG BCD pathway. As there is no chi site, so the RAG BCD pathway will join to it and will be completely destroying this foreign DNA molecule. Okay,
Correct.
there is no chi site now, that is why it has been destroyed. RAG BCD will bind, completely destroying it and finishing the process. So RAG BCD is also a mechanism to protect the E. coli from the invading pathogenic microbes as well as invading viral DNA.
A nick in one template strand can cause a replication fork to collapse, with one of the daughter DNA molecules terminating prematurely. Homologous recombination can be used to restart the replication fork.
Now, homologous recombination is a model in which we can restart the replication fork by forming complementary sequences. How? See this? Now RNAs can be degraded and the primer could be used to biosynthesize the new DNA molecule. The DNA could be sealed. Okay. Now homologous recombination can be used to repair it.
Question 2. Which E. coli enzyme would be used first to restart this replication fork?
Now, which of the E. coli enzyme will be used to restart this replication fork? It will be a RAG BCD pathway that would be used to restart the replication fork.
Correct.
Okay. It will move in this particular direction. Then RAC A is going to bind to it. RAC A will cause the strand migration to take place.
Question 3. With which strand would the Rec A coded 3' tail form base pairs?
RAC A coated pairs will form a base pair with
Correct.
this strand because this one was the complementary strand. See this? See this? This is how the base pairing will take place. Exchange. Now replication can now start here. Please look here. This is really good. See,
Question 3. With which strand would the Rec A coded 3' tail form base pairs?
it will form base pair with this particular strand because it will displace the other strand.
Correct.
Using this out, and now see this how the replication takes place. This has been replicated, this has been resolved, and now see this 180-degree rotation. This is the 180-degree rotation. Okay, even if you cannot understand this out in this particular manner, you just can also pause the lecture and could effectively visualize at every stage. Okay, the same diagram is also given in your Pathfinder book. You can also consult the same. I would be dealing with that in the evening class.
DCM1 is a eukaryotic protein used during meiosis to match up homologous chromosome pairs at the beginning of crossing over. Question 4. Which enzyme of the Rec BCD pathway has a function similar to that of DCM1?
Now, which enzyme of the RAG BCD pathway has this function similar to that of DCM1?
Correct.
RAG A was the one that was functioning in accordance with the DCM1.
Cells use homologous recombination to exchange genetic information between DNA molecules. The mechanism of recombination in E. coli is the Rec BCD pathway, which requires a double-stranded break in one of the DNA molecules involved in recombination. Several enzymes are involved in this pathway.
RAG BCD enzymes. This is just a conclusion, just a summary.
Rec BCD processes DNA to generate 3' single-stranded tails.
This tail will migrate further
Rec A promotes strand invasion.
Okay. Then
The RUV-AB complex catalyzes branch migration.
it is the one that will be catalyzing the branch migration to take place. Once the branch has been migrated, then
Finally, RUVC catalyzes Holliday Junction resolution.
this is the resolution of the Holliday junction that has taken place. Moving forward.
The double-strand break repair model of homologous recombination differs from the Holliday model in several respects. First, the double-strand break repair model involves a double-stranded break in one DNA molecule, rather than single-stranded nicks in both DNA molecules.
Rather than a single stranded nick, there is a double stranded nick which is being produced here.
Second, the double-strand break repair model involves processing of the DNA to generate a 3' tail and synthesis of new DNA, features lacking in the Holliday model.
Okay. Next.
Third, the double-strand break repair model can involve two Holliday junctions rather than just one.
Rather than just one, it can have the two double stranded DNA models. Next.
The double-strand break repair model is used by both prokaryotes and eukaryotes to exchange genetic information. This pathway can be used in several different cellular processes. For example, E. coli can use Rec-BCD to destroy foreign DNA molecules.
Okay, so this pathway can be used in distributed
Organisms can also use homologous recombination to restart stalled replication forks.
So if DNA replication is stalled or stopped, then this process could be effectively used. This is all over about the homologous recombination. That's all.
Homologous recombination is also vital for pairing of homologous chromosomes during meiosis and for crossing over between these chromosomes. You have completed this exercise.
That's all. We have completed this exercise, and here we end this particular class. We will meet very soon in our next lecture. Till then, goodbye. Thank you. Thanks a lot. Hope you would have understood this particular lecture, this particular class. Thank you. Thanks a lot.